welcome back to this afternoon session of day 2 of International Symposium in Industry, Agriculture and Healthcare, jointly organized by IIT Kanpur and IPS Waseda University. So our next session in today's schedule is a special invited talk by Dr. Anirban Chowdhury, and Dr. Anirban has joined us in the meeting. So Dr. Anirban Chowdhury is a lecturer in Neural Engineering and Robotics, School of Computer Science and Electronics Engineering at University of Essex, and also a member of Brain-Computer Interface and Neural Engineering Group at Robotics Group in University of Essex. Prior to joining University of Essex, Anirban was a postdoctoral research associate at Northern Ireland Functional Brain Mapping Facility at Ulster University, Northern Ireland. He holds a PhD in Mechatronics from Center of Mechatronics at IIT Kanpur, India, and Tech in Mechatronics from School of Mechatronics and Robotics at IIEST Shibpur, and BTech in Electronics and Communication Engineering from Kalyani Government Engineering College, India. He has contributed to 2 UK-India thematic partnership projects funded by the British Council and Department of Science and Technology. He has also led successful completion of 2 clinical trials on post-stroke robot-assisted neurorehabilitation, one in India and other in UK. His current research interests are in areas of robotic rehabilitation, brain-computer interfaces, assistive technologies, human-robot cooperation, and autonomous mobile robotics. So, welcome Dr. Anirban. Today he is going to talk about affordable brain-robot interface guided home-based telerehabilitation. Right.
Thank you very much, Aniruddha, for your kind introduction. So I can share my screen to start the talk. Hello and welcome. Today my talk is on affordable BCI, that is brain-robot interface guided home-based telerehabilitation. So first, I would like to start with what is brain-robot interface. So this is a new technology, not so new, I would say, maybe a couple of decades now. This technology is quite advanced at the moment, where we acquire brain signals and try to control robots for different applications. There are many application areas for this technology. For example, there are a few applications in motor rehabilitation. So patients who lost their limb functionality after stroke, we can use these brain signals to decode their movement intention and help them performing different activities. We can also use these signals to control a wheelchair device or a robotic arm to replace these motor functionalities if those cannot be rehabilitated. Also, we can use to restore their functionalities. For example, we can implement a functional electrical stimulus device directly on the muscle and use these brain signals to activate the muscles directly. So we can bypass the usual neuronal pathways between the brain and muscle and create an alternate route from brain to the muscle to activate the muscle signals. Then it can be used to enhance the performance of the human beings. For example, when some people are driving their vehicles and they fall asleep due to boredom, the signals can be picked up from the brain to make them alert, so enhancing their performance of driving. Recently in our lab, there is one experiment going on where they are trying to improve the performance of Formula One drivers using some sort of electrical stimulation called TDCS, or transcranial direct current stimulus. And also we can supplement the performance. So so basically, if we need some kind of compensation for our performance, or uh, for example, we can create an architecture of human robot cooperation type of work, and use these brain signals to help the robots assist ourselves in a better way. So how these brain computer interfaces work is that we put some electrodes on the brain, not directly on the scalp, not directly inside the brain. We basically use EEG signals. So we put electrodes on the scalp, and then record the signals using multiple channels. And after the signal acquisition, we do some sort of feature extraction. These are basically time frequency features that we use traditionally. And these days, we also use deep learning based architectures for end-to-end learning. And then these features are then classified using traditional classifiers like support vector machine or linear discriminant analysis, or we can use some advanced classifiers like the neural networks, convolutional neural networks, etc. So little bit into the neuroanatomy of our brain signals. As we know that there are two hemispheres in our brain, left and right hemisphere, and basically our motor movements are controlled contralaterally, means the left hand left hand motions are controlled by the right hemisphere. And this homunculus map shows the areas on the motor cortex where different limb movements are controlled. Similarly, we have somatosensory cortex for getting the sensations from our limbs. So as we know that there are two types of neurons, which one which conducts the signal from the brain to our limbs, the efferent pathways, and then the afferent pathways, that is which carries the sensation from the limbs to our brain. So you can see that the location of the hand and then leg movements. So towards the central part of the brain, you can see the leg motions are controlled there, and a little bit left and right, you can see the hand movements. So this is one of the examples from a very maybe it's one of the first studies on stroke rehabilitation using brain-computer interfaces. So this study was conducted in Ulster University in Northern Ireland by Professor Girijus Prasad. And what he did is that he trained the patients to perform some kind of a hand movement. So for example, if you look at in this video here in the bottom left side, a patient is asked to do some kind of hand movement. So like they are opening and closing their fingers in the right hand. And they trained a BCI system using these motions. Basically, what they did is that they didn't do the actual motion, but they imagined the motions, which is called motor imagery. So you can see here motor imagery. And this is the imagined movements in the brain. And they followed the signals from different electrodes on the scalp, and then they tried to make a classifier to decode between left and right hand movements. And there was a computer game on the screen. So this is the timeline of that of a single event of the game. So first 3 seconds are the preparatory stage, where subjects are asked to be prepared for a ball to appear on the top of the computer screen. And then after 3 seconds, they show a target basket. So this green basket is the target basket, okay, and the red one is the non-target basket. Now, when the ball appears on the screen, depending on whether the target basket is in the left or the right hand side, they mentally perform the hand imagery. Okay, and then the task of the computer is to decode these brain signals and drift the ball according to that mental imagery. Okay, so if they do the left hand motor imagery, when the target basket in the left hand side, you can see the ball gradually drifting towards left and falling there. In this case, it is in the right side. So the target basket was in the right side, and the ball now here it is in the left side. Okay, so it can appear randomly on the screen, and the subject or the human being has to perform these motions mentally, and then they can control the movement of the ball on the computer screen. So this shows how we can use this brain signal to control some sort of activity on the computer, and similar activity can be channelized to a robot. Okay, so in this case, this is just a brain computer interface. But if suppose this computer is connected to a robot, and this left and right movements of that ball is shifted to a robotic movement of some robotic arm or so, then it becomes a brain robot interface. Now, this is another study where you can see an example of a brain robot interface. This study was conducted in Regency Hospital in Kanpur only. So you can see this is a clinic, actually a small room, doctor's room inside Regency Hospital, where this is conducted in 2016 during a UKRI project, UK India Thematic Partnership Project. And we basically called around 4 to 5 stroke patients to participate in an experiment for around 10 sessions, where they performed some hand exercises using brain signals. So you can see that there are a lot of electrodes in the brain. There are around 12 electrodes are there, and also there are some electrodes on the muscle in the hand. Although we at that time didn't use this EMG signals, we only used EEG signals. And then some stimulus was shown on the computer screen to instruct them when to perform their disabled hands motor imagery. So this is a stroke patient, so their left hand side is disabled by that stroke. Okay, and consequently they cannot move their fingers. So we use this exoskeleton device, robotic exoskeleton device, to assist them performing the hand opening and closing movement. Now, when the stimulus appears on the computer screen, they think about that movement, and then the computer decodes the signal to see whether the features associated with that particular movement is appearing in the brain. And when we see that the features are appearing in the brain, we instruct this robotic exoskeleton to open their fingers. Now you can see, I can play the video. So you can see they are instructed to do the left hand movement. So when they are imagining of their opening of their hand fingers, you can see there are two feedbacks they are getting. One is the feeling or sensation of the hand movement using the hand exoskeleton. So their hand is being opened gradually. And similarly, they can see a visual feedback. So you can see that a hand grabbing a remote, and then it is gradually opening. So these two motions were in synchrony with each other, so that they can get multimodal feedback from both visual and the proprioceptive. We call it proprioception, proprioceptive feedback. That is the feeling of force and motion in the hand. Okay. So why there is a need to develop affordable BCI? Now, the data acquisition devices that we have used earlier were very costly. So, for example, we didn't use a wireless device. We used a wired device, 16-channel wired device. But that is also, more than 20, more than 20K, $20,000. Now, if you go for these advanced devices, which are being launched a few years back, which is called G Nautilus from GTech. GTech is a company which produces the EEG acquisition systems. They are all mostly they are all medical grade devices. And if you go for a 32 channel wireless device, then the cost will be more than $30,000, which is a huge cost. Like if you consider a low and middle income country, and also in a high income country, this price is beyond the reach of many people. Maybe some hospitals can afford it. The existing exoskeletons which are there in the market, they are also quite costly, 40,000 K. For example, you can see this glove, ESO glove from Roseco Technology, which is a commercial grade device, and it was used by patients in Singapore and some other parts of the world. But these devices are very costly, like around 40K. So the total cost of the of a brain computer interface, if you consider using these high-end devices, it will be around 70K, $70,000. Now, there are some limitations with these exoskeletons also, because these exoskeletons are pneumatically controlled, and they don't have position control or some force feedback or position feedback you cannot take from these gloves, because they operate in a open close motion. So once they are switched on, they perform a closing motion or an opening motion, but you can't control it in between or get any feedback from them. Now, what we are proposing as a solution in our new project is to go to a low end, go towards low end devices, but keeping the same or almost the same performance. And one advantage of with such devices is that it can be used for home-based setting. Okay, so if you want to do home-based rehabilitation, where you don't need an active assistance from a clinician or a technician, then these devices can be very helpful. Now you can see that we are using the Muse S device or Unicorn device, Unicorn hybrid black device. This is also from GTech. Muse S from a different company, but these devices cost very low. So, for example, Muse S is around $300, and if you go for Unicorn, it is $2,000 around, including all the Python packages and everything. So as compared to a 30K device, this is very low cost. But also you need to consider that you'll get you need to consider the signal quality and also the number of channels. For example, Muses is only 4-channel device. Unicorn is 8-channel device, and you may or may not get electrodes in the proper places in the brain. So for example, for controlling motor activities, we need electrodes on the motor cortex. Now, you may or may not get the very high density electrode arrangement on the motor cortex. Now, for exoskeletons, so we already built this exoskeleton back in 2015 or 16, which was used in 2 clinical trials before. And this is a very low cost exoskeleton which can be manufactured under $1,000. And this is also 3D printed, so we can quickly manufacture it using any low cost 3D printer, and it is very easy to assemble. So thus the total cost of the device can be scaled down to $3,000 only. Now see a drop of around 95%. So with a cost of 4.2% of a high-end device, we can produce something under 3K, which can be used in a home settings, and it can be wearable, easy to use, and it can be used for telerehabilitation applications. So that was the first challenge, how to use these low-cost devices to get similar performance as compared to the high-end devices, and then go for the telerehabilitation applications. Because unless we solve this problem, we cannot reach for a home-based physiotherapy, because the high density devices are very difficult to set up without proper technical assistance. It needs electrolyte gels and everything, which is not possible for a novice caregiver. So creating the software for the BCI or the BRI. Now, we need applications for data acquisition, applications for showing stimulus on the screen, applications for signal processing and machine learning to decode the brain signals, and applications to show the visual feedback on the screen for controlling the exoskeleton. So these are the few challenges we need to meet using these low-cost devices. So first is the data acquisition, how to get the data. Then we need two things. One is the EEG signals, which we need to process, and also we need to show the stimulus on the computer screen. And these two has to be in synchrony with each other. So there should not be any time lag between these two things. And concurrently, we need to decode these signals and show the visual feedback and the exoskeleton feedback to the user, which also has to be synchronized with this signal processing. Now, how to do this setup? So first, the data acquisition. Now, a standard protocol to do this data acquisition from these devices is using lab streaming layer protocol. Okay, so this is a very effective technology which can be used to time synchronize the signals acquired from different devices. So for example, we have an EEG device, Muses or Unicorn device. You can have an eye tracker, like a Tobii Pro Nano eye tracker, or a EMG acquisition signal. And from all these sensors, we can create something called LSL stream, lab streaming layer stream. Okay, and this can be done using, there are several Python packages to do this. Basically, these devices sends the signal through via Bluetooth, and then we need to receive these signals wirelessly in our computer and then create these streams. Now, for Muse, we have something called Muse LSL. So this is a Python library. If you run a simple command after connecting your Muse device, then you can start this LSL stream. Similarly, for Unicorn also, we can create similar stream using a particular API that Unicorn provides along with it. Now, we can create similar streams from the event markers also. So event markers are required along with the EEG signals to know what is happening in which time. So, for example, we need to know when a particular stimulus was shown on a computer screen, and where it fall on the EEG signal timeline. Now, for these type of setups, we use the PsychoPy library in Python. So this is an open source library to create stimulus, and we can similarly create an LSL stream for the PsychoPy also. Now, the stimulus stream and the sensors then can be fed into a particular API called lab recorder, and then the lab recorder can synchronize all these signals coming from different sources and give the same timestamp, and then it can be stored in a particular format, which is a standard format is XDF file format, and this file format can be readily used in some some commercial, open source EEG analyzers, such as EEG Lab. So if you directly feed these signals into EEG Lab, it can show you different power spectrum densities or time frequency plots, etc. So this can be used for offline analysis and can also be used for online processing. Now, coming to this stimulus generation, how do we generate the stimulus? So we use PsychoPy, which is a free cross-platform package allowing you to run a wide range of experiments in the behavioral science. So this was developed for basically psychology experiments, but this can be also used for brain-computer interface, because there are some similarities between psychology experiments and BCI experiments, because we need to instruct the participants in both of these fields of science. Now, it's an excellent tool to create simple non-VR based experiments. So you don't need a complicated virtual reality platforms to, show the stimulus. You can use simple schematics using this PsychoPy device, which can show some simple instructions, some very low-level graphical instructions to instruct the participants, and that would be quite okay. So you don't need more than that for a basic BCI experiment, unless you go for a very enriched feedback using VR. So using PsychoPy in conjunction with the exoskeleton allows us to gather a lot of data from healthy volunteers and refine our signal processing and interference and inference algorithms. Base experiment created and tested, including extra multi-threaded code to the package to allow concurrent EEG measurements. Now, but we need to be careful that these two signals, the EEG acquisition and the stimulus generation, should run in two different threads, so that they can be concurrent. They can be concurrent to each other. Otherwise, there would be a lag in the device, and then it will jeopardize the whole aim of the system. So this is a screenshot of a PsychoPy toolbox. So one of the big advantage of using PsychoPy is that we can use this builder view of the toolbox to create the experiment using a graphical user interface, and then a Python code can be generated directly from these configurations, and we don't need to write these codes. Okay, so once we configure the experimental paradigm, for example, you can see we have designed an experimental paradigm here. So this is the start of a experimentation event, and there is a wait period, and then there is a get ready period to instruct the subjects to get ready for the stimulus. And then there is an action routine. Action routine means what we are instructing the participants to do, maybe left hand imagination or the right hand imagination. And then there are some further instructions. And this loop here tells that this routine will repeat for several times. It may be 40 times or 50 times within an experimental run. Okay. Now, after creating this environment, we can create the corresponding Python code for it, and then we can integrate our EEG acquisition, data acquisition code directly into this Python code. So thus we can merge the EEG acquisition and the stimulus generation together in the same Python code, and you need to run only that Python file to do both the stimulus showing and the EEG data acquisition. You can also write the signal processing code into it. Now, if you go for online signal processing, then all the online signal processing code can be written inside it, or we can use it simply for data storage. So after the end of the experiment, the data can be stored in a XDF file, and then it can be later used for offline processing. So how to you know uh, so this is the first step we need to do before going into the experiment when we are using the Muses device. So for Muses, there are two approaches to acquire the data. One is when we are using a Windows machine, we can use this small program called BlueMuse, and what BlueMuse does is it creates a LSL stream. So once we connect the device, switch the device on, and we open this BlueMuse application, then it shows all the Muse devices connected to the PC, and then we can select a particular Muse device and start the stream, and then the streams can be shown here. Now, Muses has several other sensors also, like accelerometer, gyroscope, PPG, telemetry, etc. And you can see the timestamps and the sampling rate here. Now, for EEG, we have a sampling rate of around 256 hertz. And alternatively, if BlueMuse cannot be installed in the PC, then we can use another Bluetooth dongle, which is BLED 112 Bluetooth dongle, which costs only $15 or so. Now, this dongle, after using this, putting this dongle into the PC, we can install the library called Muse LSL, and then we just need to run a particular command in command prompt in the Windows, and this will create the same LSL stream which is created by BlueMuse. So there are two options to get the data from BlueMuse. And what is important to note that what we are creating here is the LSL stream. Unless we create this LSL stream, it is difficult to get the signal directly into the Python file. Now, integrating Muse's stream with the PsychoPy. So PsychoPy can generate the Python code for the generation of the event signals or the stimulus that we discussed already. Now, in this code, we need to get the Muse LSL stream containing the EEG data. So we already have the PsychoPy code, which is generated from that GUI. Now, what we need to do is to integrate the Muse LSL library with this code so that we can get the Muse LSL stream containing the EEG signals directly into the Python file. So we can use another library here, which is called the PyLSL library. And just to then we just need to run a few lines of code. For example, we need to create the stream, first of all, this stream object using the PyLSL library. And here we basically specify the particular type of stream that we want to acquire. So here EEG means we are trying to acquire the EEG stream, and then we define how many chunks of data we want to collect. So in this case, we are collecting 12 chunks of data, and if there are several EEG devices, we can actually we can uniquely identify them using this stream array. So here, if there are multiple streams, so maybe there could be stream 0, stream 1, etc. Now, if there is only one stream, then it will definitely be stream 0. Okay. So we create this object inlet from this function, from this method stream inlet, and then this inlet object will be used to get the data. So now you can get the EEG data and timestamps both using this inlet object by using this method called inlet.pullchunk. And then you can also define how many the length of the the shift of the data. So when you are getting the data in some chunks, what will be the time shift between them? Okay, So we can define this time shift. Typically, for example, we can, depending on how fast we want the system to behave. For example, in a typical BCI experiment, if we can set a time shift of 100 milliseconds, that will be sufficient. So we can fix this time shift length as 100 milliseconds or so, and this is the fs is the sampling frequency. So if you multiply the sampling frequency with a shift length, then that much of data you will get at one time. And then the draw EEG signals from all the channels and the timestamps corresponding to each sample can be stored. And it can be directly streamed into your Python code. So in this variable, EEG data, you will get chunks of EEG signals at each at each shift, and then we can process the data. And the data processing code actually works quite fast. So if we keep the shift length sufficient, if we can keep sufficient shift length, for example 100 milliseconds, it will be more than enough to process the data, because for processing maybe we need 50 milliseconds or so. So by the time the next chunk is available, you can process and decode the signal. So so here I'm giving an example of a typical BCI experiment, so which was used in a clinical trial previously in Kanpur and then in Northern Ireland. So there are 50 such trials in a particular experimental run, and one trial runs from 0 to 8 seconds. Now, for the first 3 seconds, we show a fixation cross, like a small cross in between the computer screen, to instruct the participant to get ready for the cue. Cue is the instruction that we tell them to do. Now, it can be a left hand imagery or a right hand imagery, or it can be a hand grasp action or a rest. So for example, if we during the cue period, if we show a cue to hand grasp, then they will mentally perform a hand grasping motion. Yeah, and if we show rest, then they will simply relax and do nothing. Okay. And then after 1.5 seconds, we start giving the feedback, the neurofeedback. So we'll simultaneously process the signal and show the corresponding output in the computer screen. So this is the feedback shown at different timestamps. So from 4.5 to 8 seconds, if they can hold this motor imagery continuously, and we get the right class, we will try to decode the right class, then there would be a full opening of the hand, and correspondingly the exoskeleton will also open the hand gradually. And these are the two markers, or the event markers, that I was talking. So this event marker is related to the trial. So it becomes 1 when the trial starts, and then again becomes 0 when the trial ends. And then there is a event marker for the label. So label shows which class it is, whether it's a left hand imagery or a right hand imagery or a hand grasp or a rest. So there are different labels associated for different classes. So for example, 1 is associated with class 1, 2 is associated with class 2. So this signal becomes transitions from 0 to 1 when there is the class 1, and this level becomes 2 when it is class 2. So this is how we can distinguish between the two classes, and then correspondingly we can process the signal to decode it. Now, this is the neurophysiology behind what happens in the brain when we try to perform a motor action. So this is the power spectrum. You can see that between 8 to 30 hertz, that is which is called the sensory motor rhythm. And during this signal band, we can see a drop in the signal power. So this is when they are not doing anything. You can see the signal power. If you see this line in between, the signal power is above a particular threshold. Okay, and when they try to perform a movement, then you see that this level drops below a particular threshold. Okay, and this drop can be seen in the timeline here. So this is the baseline period when they are instructed to be ready for the stimulus, but the stimulus was not shown. Now this is the movement onset, when you can see a drop in the signal power, and then it comes back again after a few seconds. Sorry, after a few seconds, yes, 1 or 2 seconds, when the movement ends or the motor imagery ends in the brain. So this is the particular signature we try to look when we try to decode the movement related EEG signals. Now, just to talk about the signal processing pipeline which we use in our experiments. So the EEG signal is first segregated into trials. So first there is a continuous recording, and from that continuous recording we break it into several trials. So suppose there are 50 trials and the signals are acquired from 4 channels. We have 4 channels, so in this case this is 4, and each trial consists of 2048 samples. Now, assuming that the trial length is 8 seconds and the sampling rate is 256 hertz, we get to this number 2048. So this is the entire data set that we get as EEG signals. And then we also have the labels corresponding to each of these 50 trials. So suppose we have some hand grasp trials and some rest trials. So they are equally divided into 25, 25. Okay. Now, so we have the raw EEG signals and we have the labels. All we need to do is to extract the features and then train a classifier. So first we do the bandpass filtering for each of these trials, and the filtering can be done in 2 frequency bands, 8 to 12 hertz and 16 to 30 hertz. So 8 to 12 is the mu band and 16 to 30 is the beta band. So after doing this temporal filtering, then we need to do some kind of spatial filtering. So why we need to do spatial filtering? Because all the channels are not equally important for the signal processing. So there can be many channels which are not relevant or partially relevant. Now the class features has to segregate these two categories and only take into account where there is a greater distinguishing. There is a greater difference between these two features corresponding to this the class 1 and class 2. Now for spatial filtering we use something called common spatial filter. The size of the CSP filters are of dimension number of channels into number of channels. So for example, if you have 4 channels, then using the raw data or the data acquired from the training trials, we create these WCSP matrices which are of the dimension 4 cross 4 if you have 4 channels. And after creating these WCSP matrices, in order to generate the feature vectors corresponding to each trial for mu and beta band. So we pass these signals through this WCSP matrices. So basically we multiply the data acquired from a particular trial, either bandpass filtered EEG data with these WCSP matrices and take the log variance of it. And after doing the log variance, you'll get a vector equal to the length of the number of channels. So if you have 4 channels, then you'll get a feature vector of 4. Now, after doing this log variance, we need to take the first and last elements of the feature vector, because there are greater difference in the feature pattern when you use the terminal elements. Okay, so you can take first few elements from the feature vector and the last few elements of the feature vector. So for example, if you have 20 channels, so then you can use first two elements and the last two elements of the feature vector. Now this happens for every frequency band. So if you have two frequency bands, then 2 into 2, 4 feature elements will get. And if you are using 50 trials, then the size of the feature space will be 50 cross 4. And now this 50 cross feature space, along with 50 labels corresponding to each classes for all these 50 trials, we need to pass it through a traditional classifier, for example, support vector machine or so, to create the decoder. Now, if we look at the feature pattern, this is the first best feature and the second best feature. So first best feature is the first element of the feature vector, and the second best feature is the second element of the feature vector. So all these plus signs and these small round signs basically shows the features from the two different classes. For example, if we do the left hand and right hand motor imagery, so these the distinction is between the colors. So the blue one shows the right hand motor imagery, and the red one shows the left hand motor imagery. So okay, sorry, my bad. Let me explain it clearly. So here we are actually showing the features, shift in the feature between the two sessions. So suppose we have a training session and a feedback session. So we first do the training session and then we do the feedback session. So this red one is from the feedback session and the blue ones are from the training session. And then we show the two different features corresponding to the left and right hand motor imagery. Now, briefly, I would like to talk about the process of giving online feedback. So as we discussed, we get the EEG signals, do the data acquisition and with class levels, and then we do temporal filtering, spatial filtering, and training the classifier. And after training the classifier, we repeat the same process in the online feedback, but then we need to do it like chunk by chunk. So whenever we get a new chunk, we process the data on this new chunk and feed the features into the classifier to get this feedback, visual and experimental, visual and exoskeleton feedback. So what we are proposing with all these background signal processing is to use either Muse or the unicorn device, which can be connected via Bluetooth, and the entire signal processing will be done in Python using PsychoPy and this CSP plus SVM classifier, which can be processed in a low-end computer only. You don't need a high-end computer, and then it will be wirelessly or connected. That is, Bluetooth connection will be there with the exoskeleton device to control it. Now, I would like to discuss the difference or the improvements that we got from the earlier trial. So previously we used this 16-channel device, which was more than 20K, and now we are using the low-cost devices, which is under 2K only. These are dry electrode systems, so it is easy to set up. It can be set up within 5 minutes, where in the previous system it took 15 minutes to set up. And these are all wireless, where previously it was wired. So this is highly portable, and it can be processed in any standard lab. So yeah, so this figure is misleading. Sorry. Any standard laptop which costs around not 40K, it should be 400 dollars only. 400, not K, please ignore this K. It should be $400. While for the high configuration workstations, it was around I think around in Indian rupees it was 2 lakh. Okay, so please ignore this figure, it was wrong. So basically, previously you need high configuration workstations. Now it can be processed in a standard laptop, and this time it is fully open source using Python. And previously we used MATLAB, so it's you can get more freedom in this case. And wireless connection between laptop and exoskeleton. Previously it was a wired connection with the laptop and the exoskeleton. And if we compare the accuracy differences, so with the high-end device, we got an accuracy between, so we started with an accuracy of around 65%, and then it went up to 80%. While using this very low-cost device, we are at 70% at the moment. But this accuracy will improve when we'll use some deep learning architecture, which is the current trend in the EEG technology, and upgrading these muses to the commercial grade GTEC device, which is the unicorn, which has some channels on the motor cortex. So definitely this accuracy should improve from 70% to, for example, 75 or 80%. And this is the feature plot we got from the muses. So you can see the distinction between the two classes, class 1 and class 2. And here it appeared in alpha band 9 to 15 hertz and the beta band 20 to 30 hertz. We have also noticed ERD, event related desynchronization. So you can see this drop in the signal power when the participants did motor imagery, and it appeared in a particular channel called TP9, which is ipsilateral to the hand movement, which is a bit counterintuitive because generally it happens in contralateral hemisphere. But for slow finger movements, it is observed in previous literature also. So this actually shows that we are able to detect the neurophysiological features from these low-end devices also. And this is the demo that we did recently. So you can see this participant wearing a Muse's device, and they are performing the motor imagery, and they can wirelessly control this exoskeleton device. So you can see that this is not connected with the wire, and the whole system is wireless. Now, in an ideal scenario, this exoskeleton will be owned in the other hand of the patient, on the disabled hand of the patient. And this is the telerehabilitation architecture which we propose to develop. So all these exoskeleton and BCI system will be based on a home setting, where the caregiver will be in charge to put these things on the patient, and the data will be uploaded wirelessly to a computer app in a mobile app in the hospital site. So for example, in India, if there is a hospital in the urban setting and there is a rural environment. Now in India, you know that even in villages we have good internet connections these days. So I think this kind of architecture can be realized. So using an app, sitting in an urban hospital, the doctors can control, intervene in the therapeutic process and also monitor the progress of the patients sitting at home. So they don't need to come to the hospitals. It will be quite cheap and affordable. They don't need to pay for the regular physiotherapist visits, which is very costly. So this is the overarching aim of the BRIGHT system, the telerehabilitation system which we propose. And right now in Cambridge, we are trying to do some clinical trial to establish the feasibility and the clinical efficacy of the system. And we designed these experiments using single case experiment design. So we'll establish a baseline using some motor outcome measures. And after the intervention, we'll measure them again to see the difference. So this is the whole planning to get 5 measurements before and 5 measurements after of their motor ability, and giving 16 sessions of physiotherapy in between to see the changes in the motor improvement. So we are hoping for the validation of our system in future. So these are the list of publications which somebody can follow to get more information. And thank you. Thank you very much for listening to the talk. And I'm open to any questions.
Yeah, thank you, Dr. Anirban, for that insightful talk. So I would request some questions from audience. You can raise your hand if you have any questions. You can direct it to Dr. Anirban. Okay, so I have a question.
Yeah.
So regarding this telerehabilitative device that you have demonstrated, so how does the feedback from the patient's hand is taken, like the force feedback, if it is captured in the device?
Yes.
So is there
Yes. So the previously we did two types of feedback. One is the force feedback. So there are some force sensors in the finger mounting caps. So if I can show you the device. So if you look at this device, there are some black tapes inside the finger mounting cap. So there are some force sensors. So you can get how much force is acting in the flexion and the extension direction. So you can give it as a feedback to the patients. And also the movement of the fingers, it can be shown on the computer screen. So so I have shown, you can see this sequence of images. So these images are connected with this motion of the fingers. So as the fingers are opening, they can see this virtual hand on the computer screen, which is opening. So both kinematic feedback and the force feedback you can show to the patients.
Thank you. Thank you. So is there any questions from the audience? So,
sir, may I ask a question?
Yeah.
Okay. In the video you have sorry, in your presentation you showed that the two sides of our brain, like left side controls the right side and the right side controls the left side. And in the experiment at the last, you showed that the person is controlling his hand with easy signal, and the prototype is on the on the prototype you have shown it will be worn by the person in his on his other hand. So
yes, let me explain this thing in a better way. So right now you are seeing that this gentleman is wearing an exoskeleton in the left hand. Okay, and there is another exoskeleton which is there on the table. Okay,
yes.
Now this we did just to show that the human is not deliberately moving their fingers. Okay, they are just doing the mental imagery in the hand, and the exoskeleton is actually controlled by the computer. So this is for the demonstration purpose only, so that the people can appreciate what's going on. Now, in an ideal setting, when the patient will be wearing this exoskeleton, then there will be no, there will be no extra exoskeleton there. So this will be this exoskeleton will be worn in their disabled hand. So suppose they are paralyzed in the left hand side. So they will be wearing the exoskeleton in the left hand and then perform the motor imagery in their head. And then when the signals will be decoded, they will get the feedback in the left hand only. So the architecture will be similar to what I have shown in the first slide, towards the beginning. So this will be ideally like this. So it will be shown in the computer screen. The stimulus will be shown. They will be performing the motor imagery, and then you see the movement in the hand, left hand finger and on the computer screen. So this will be the architecture, but previously it was all wired system and a very complicated one, and now it's a very low cost, affordable and easy to use. So that's the difference we are trying to make for home-based rehabilitation.
Thank you, sir. Thank you for your answer. Your presentation was very nice and enlightening.
Thank you.
So thank you, Dr. Anirban, for that wonderful talk.